Welcome to Chapter 3, Part 1 of Anatomy Physiology 1. We're going to talk about cells, cell membranes, and osmosis, to name a few things. I thought this was really interesting. Here's a series of cartoons that I ran across. Biologists are a bunch of cells talk about other cells. It's interesting that our cells can be so different that some of them can make us think about what we want to say. Some of them can make us have a tongue and a resonating pharynx for our words to come out of. So anyway, I thought that was interesting. Another thing that most people don't realize is how much of our body is composed of bacteria and viruses and fungus. So you've been taught since you were a little kid that viruses are terrible and bacteria are terrible. But hopefully by the end of this chapter, I can convince you that not only are bacteria not terrible, but we actually can't live without them. So that's one of my goals. I'm not sure about this statistic, but it says 57% of your body is made up of other living beings and other life forms besides you and your cells. So you're only 43% cells. And here's one you have to stop and think about. Biology is the only science where multiplication and division are the same thing. So when a cell multiplies, then you go from 1 cell to 2 cells to 4 cells to 8 cells. So you're multiplying, but the way you do it is you divide the cell into 2 cells. So things to make you stop and think and go, how about that? In the last chapter, we said that all living things are made of cells, and here we are in the next chapter saying all organisms are composed of cells. So that must be a pretty important concept if we keep talking about it. If you can understand how the cells go together and how they work, you can figure out some of the things, how our body works and how diseases happen. What goes wrong with cells that cause us to have a disease condition? And then how can you fix it? The rationale of the therapy. So these are the things that we hope to talk about in this chapter. It's only been a couple hundred years since we even were able to see cells because we developed microscopes and we got more and more sophisticated microscopes. And now we see all kinds of things that we didn't know were out there before. This is a picture of the microscope that Hooke used, and this is what he was looking at, and this is why he named cells cells. So we don't have cells like this. This would be some sort of a plant. But what an interesting thing to see through the microscope, and what a beautiful microscope for that matter. So I just happened to find a picture of a tardigrade taken under an electron microscope, and I just thought it was funny. What would he have called cells if this was the first thing he had seen under his microscope instead of this? So 3 of the men that we studied in the last chapter have reappeared in this chapter. Robert Hooke was the one who came up with the term cell back in the 17th century. 2 centuries later, Schwann was the guy who came up with the idea that all animals are made of cells. And then in 1859, Louis Pasteur proved once and for all that things don't spontaneously generate. So before that time, people thought that life just appeared magically. And if you left out a bowl of beef broth and you came back the next day and it had maggots in it and it had worms and had mold growing in it, then that was proof that life just arose spontaneously. It just appeared from nowhere. And of course, now we know that the air is full of spores and they can land on a wet, nourishing surface and start growing. So Pasteur did an experiment where he took some beef broth and he put it in a container that was open to the air, but it was upside down. So any bacteria, any fungus, anything that could infect the beef broth would have to go up through the opening and then through a kind of a convoluted path to get into the beaker and then start growing. And so he left the beef broth out and nothing grew in it. And so that proved that it doesn't just spontaneously generate, that there's something in the air that causes things to go bad. Some of the components of the cell theory are that all organisms are composed of cells. The cell is the simplest structural and functional unit of life. All of the organism's structure and functions are due to activities of cells. Cells only come from pre-existing cells, and cells of all species exhibit biochemical similarities. So I agree with pretty much everything in here, except for cells come only from pre-existing cells. And here's why. In this chapter, we're going to talk about the cell membrane, and then we're going to talk about the organelles that are inside of the cell membrane. And here's an article that they published in Astronomy about the cell membranes, and they're actually finding those molecules in outer space. These things are self-assembling. If you put them together, they will form a sphere. They will go around and turn into a spherical shape or a globe shape. So if these are out there in outer space, then they could spontaneously come together into an empty cell. Now, in this textbook, they don't talk about it, but in some of the earlier biology textbooks, if you've taken like general biology, they should talk about a graduate student who was looking at the different gases that were in the earth before there were cells, before there was life. There was ammonia, methane, a little bit of water, and he took the gases that existed in the early pre-life era, put it in a sphere, and shot it with electricity. And some brown tarry stuff formed on the inside of the globe, and when he opened it up and he analyzed it, he found that amino acids had spontaneously formed from just electricity and simple gases that were around when the earth was very, very new. So we know that amino acids can spontaneously generate with a little help from lightning or some energy source. And we know that there are cell membrane molecules that they're finding in outer space. So that's why I have a little pause when it says that cells can only come from pre-existing cells. Because, there had to be some cells in the beginning to have the cells come from, if that makes any sense. Now this is going to be interesting. We'll not talk a lot about this because this is human biology, human anatomy. But if you look inside of worms, if you look in dogs and you name it, what animal that you want to think about, you're going to find a lot of the same chemicals. You're going to find a lot of the same enzymes. So it's repeated throughout a lot of the life forms. And if you start looking at plants, then you're going to find more biochemical similarities. The same chemistry going on, basically. So one of the times I had some students, we were playing around with the database. So every time you find the structure or the DNA sequence of a gene, you can put it in this database, and then people all over the world can play with it and look at it. And so one of the students was just being silly and they pulled up the instructions, the genetic information on how to make hemoglobin. And then they pulled up a tree and started looking to see if there was any hemoglobin in trees. And I started to say something like, they have chlorophyll and we have hemoglobin. And the kid got a hit. So trees actually have the genetic information on how to make hemoglobin. So there is a lot of biochemical similarity. I hope in lab that you spend the time playing with the microscope and looking at the different cell types. So you've got cells like the squamous cells that make up our outer skin and that line our blood vessels, that make up the alveoli of our lungs, and they're squished flat. So you need stuff to get across from one side to the other quickly. So a flat structure works best for those particular cells. Cuboidal are just what they look like. They make a cube, like a dice. The perfectly square on all 6 sides. Anytime you have cuboidal cells, you know that they're going to be absorbing or secreting something. That's kind of what they do. The columnar, these are going to be tall, skinny cells, and we use them for absorption. And then nearby, wherever you have columnar cells, you often have mucus secreting cells. So you've got these cells absorbing, and then you have mucus to move stuff along so you can get more stuff to absorb. You have mini shape. Polygon means mini shape. The prettiest one that I can think of is a nerve cell. It kind of looks like a very beautiful kite with a long tail. You have star-like cells that radiate out, and a lot of those are in the brain, and they help connect the neurons up in the brain. You have a lot of round or oval shaped. So usually when you think of a cell, you think of a round cell. But actually there's not a whole lot of them that are round. The red blood cells and the white blood cells are round, but they're free to stretch out because they're floating around in plasma. Most of the other cells are bumping up against other cells, and that kind of constrains the way that you can shape. Some are disc shape. Some are fusiform, so they're thick in the middle and pointed on the ends. And a lot of them are fibrous. I always have my students imagine getting a pot roast and then putting the fork in the pot roast to eat it, and it comes off in fibers. It comes off in little stringy pieces. And you're going to find out that muscles are fibrous. They have long, skinny cells rather than the round cells that we normally think of. So the other thing that they point out is if you're looking at a cross-section view versus if you're looking at a coronal view, for example, the cell's not going to look the same. So just a warning in lab when you're looking and you're trying to find like a squamous epithelium that looks like a fried egg, if they cut it on the side, it's going to be so flat that you barely can see it. Pretty much all you're going to see is a line with a little bump on it where the nucleus is sticking up a little bit. Here's just an overview. The squamous are flat, cuboidal or square, columnar are tall and skinny, and they usually either have microvilli on it or they have cilia. These happen to have cilia on them. And mini-angled, see where they bump up against each other? If you've ever made cookies and you put too much batter on it, when they cook, they all run together. This is kind of what you get. So they stop when they bump into another cell. So they have weird angles instead of being round. And here's the star-shaped ones, and you can kind of see them reaching out to touch. Spheres all your white blood cells are spherical, looking like discs. To me, these look like those lifesavers that have the center filled in, but they call them discoid or disc-shaped because they've lost their nucleus. So they've actually caved in. This is actually caved in on both sides. And here's your spindle shape. So this would be like your smooth muscle. And then there's your fibrous. There is only one cell in the human body that's actually big enough to be seen without a microscope. And that is the woman's egg. And she makes it really large so that the sperm have a good chance of finding it and impregnating it. So the only visible cell. All the other cells you have to use a microscope. And as you notice, you probably use the 4x objective to find some of the cells, and then you use the 10x objective and on up to the 40 objective to get any kind of details at all about the shape of the cell, what's inside of the cell. If you try and make a cell too big, it'll actually pop. So we're kind of limited to the size that cells can be. The egg has got a lot of protection around it to keep it from popping. For those of you who enjoy math, if you take a cell and you double its diameter, so increase diameter by a factor of 2, that means double it. Now that cell, even though you doubled the area across the middle of it, its surface area is 4 times larger. So you've expanded it so there's more surface area. So more things can come out, more things can go in. And the volume is increased by 8 times. So all of the stuff, the fluid, the cytoplasm, all of that inside is 8 times more than in this one. So it's a geometric progression. The microscopes that you use in laboratory are called light microscopes, and they go anywhere from 40 magnification all the way to 1,000 magnification. You can get specially adapted light microscopes and you can go beyond that, but that's what almost all microscopes go for. Anywhere from 40 times bigger than it is in real life to 1,000 times bigger. You need that 1,000 times bigger if you're going to be looking at bacteria. Not too long ago, they developed the transmission electron microscope, and its resolution is so much better. You can go way beyond 1,000. Here's a picture of a transmission electron microscope, and there's the part you look in, and you can see that it is very, very involved. They cost anywhere from $500,000 to many, many millions of dollars, depending on how fancy you get. So we don't have those in the student laboratories for you guys to play with. It takes a lot of training, and it takes all kinds of techniques before you can get your sample and look at it. So an advantage to a transmission electron microscope is that you can see all sorts of things inside the cell that would not be visible otherwise. But the big drawback is it can only focus on a narrow slice. So you have to take a cell and literally slice it so that you can look inside. And then you can see the nuclear envelope, you can see the pores, you can see the nucleolus. So we'll be talking about that. You'll have vacuoles. The scanning electron microscope has more realistic and easier to understand pictures, but it can only look at the outside of something. It can only look at the surface. So you just scan over. So what this is just somebody took a bunch of pollen from different plants and looked at it under the electron microscope. And if you look at this, you kind of get a feeling. It's like, Ah, now I understand why I sneeze when I go outside, especially if somebody's mowing, so they're throwing seeds and grass up into the air and pollen, or in times where the wind is blowing and you can see the pollen flying through the air. So this is a scanning electron microscope. So the best that you're going to be able to see using a light microscope, you can see the plasma membrane or the cell membrane. You can see the nucleus, and you can see the fluid that's inside the cell. And if you look inside the nucleus, you can see the nucleolus also. But that's pretty much it. Now, if the cell is dividing and the DNA has condensed, then you can actually see the DNA also. So we'll be definitely looking at that in the chapter where we talk about cell division. So you're making about 30,000 cells every minute in your body, somewhere. A lot of them are in your mouth because you have to replace those cells. Down your GI tract, lining your intestines, those are constantly being replaced. Now, I actually worked with a woman who had fingernails not this long, but just about this long. And they finally had to make her cut her fingernails off because if you stop and think about it, how do you wipe with fingernails like this when you go to the bathroom? And so they took cultures. They swabbed the fingernails and found they were covered with feces. And so it's not very hygienic. So she was forced to either quit or cut her fingernails off. But clearly this person is quite fond of their fingernails. So you're constantly having dead cells dying and pushing out to make your fingernails. Here's another picture that they put. So this is a white blood cell. It's called a polymorphonuclear leukocyte. So leukocyte means white blood cell. Polymorpho means many-shaped nucleus. So look at that. Instead of it like a spherical nucleus like we're used to, this thing has lobes like that. But you can see this under the light microscope. And you can see these bags of chemical that are in there. Now this is a picture of the transmission electron microscope. So you can see a lot more detail. This makes more sense to me. But this one, you would have to take a slice and then take another slice, and then assemble it in a 3D picture to see what does the nucleus look like. Because just looking at this slice through the nucleus, it's kind of hard to tell what's going on. But here, you can clearly see the nucleus, and that it has several lobes. This one has at least 4 lobes that you can see. So we're going to learn the parts of the cell, and you have the cell membrane, or the plasma membrane, as some people call it, and it holds everything together. It's a barrier between the outside world and the inside world of the cell. So intracellular, extracellular. Your plasma membrane is made up of proteins and fat, and most people are really, really surprised to find their cell membrane is fat. So again, people who think that you need to cut fat out of your diet, cholesterol is bad, and all of that, you can't make new cells, and you can't repair old cells if you don't have fats in your diet. So just don't eat too much. A whole bag of Doritos is too much. I'm just telling you. The $2 bag, too much. Cytoplasm comes from the word cyto, which means cell, and plasm is kind of like plasma. If you think about, you have red blood cells and plasma. Some people donate blood, some people donate plasma. So this is the plasma that's inside of cells. Inside the cytoplasm, we have tiny organelles. So you and I have organs. We have heart and liver and spleen composed of cells. But if you're just one cell big, then the little things that do the various tasks inside your cell we call organelles, or little tiny organs. Then you have a cytoskeleton, because if you're made of fat, if you're made of cholesterol and fatty acids, you're needing something to hold you in whatever shape you happen to be. One of those star-shaped, fusiform, columnar shapes. And then within the cytoplasm also are inclusions. So it's things that you're storing inside of your cytoplasm. The fluid outside the cell is called extracellular fluid, outside the fluid. And it's also known as interstitial fluid, or between the cells, interstitial fluid. So extracellular fluid or interstitial fluid. In lab, you should have learned the names of all of the organelles inside of a typical cell or a representative cell. In this particular cell, we have the outer cell surface that has the microvilli, or the little finger-like projections. So instead of just having a smooth cell surface, this one actually takes proteins and pushes the cell membrane out. And this is to allow for more surface area. So whatever's going to go across the surface of this cell is going to have to percolate in through these finger-like projections off of the cell membrane. So you're going to be able to absorb more. So without even knowing what cell this is supposed to be, I can tell you that it is going to be absorbing things, because that's why you need this. Inside, holding the shape, you have microfilaments. And then holding cells together. So here's a cell right here, and it comes up against another, or abuts another cell on this side and on this side, and that's what gives it its shape, by where it bumps into another cell. So this one is almost like an upside down crown or something. Here we have proteins that are holding these two cells together. And we're going to be talking about the different ways you can hold cells together. Some you want to hold loosely together, and some you want to hold very tight. So we'll talk about tight junctions and desmosomes and other things that hold it together. But to me, this looks like Velcro. So here you have some proteins in this cell, and they extend across, and some cells in this one, some proteins in this one that extend across, and they meet right there, just like Velcro. You have the nucleus right here, and they've cut it away so you can look inside of it and see the nucleolus. So those two words sound so much alike, the nucleus and the nucleolus. So be careful looking at those two words. They used to just think that this was something when they stained the cells, that it just was a wad of some kind of stain that got up inside of the nucleus. But they kept finding it in all the cells, and sometimes you find one, sometimes you find 2 or 3 or 4 of them. And they realized, oh, wait a minute, this is something. And later on, they took some of the nucleolus out and analyzed it, and lo and behold, it's some RNA. So usually when you think about the nucleus, you go, the nucleus has DNA inside of it. And DNA is what makes up genes, and genes are found on chromosomes. So you just usually associate the nucleus with DNA. And here we have a wad of RNA, and it is making the RNA for ribosomes. Now, here's the tricky thing. I want some things to leave the nucleus, like the ribosomal RNA needs to be able to leave so that you can make ribosomes out here in the cytoplasm. So there are holes or nuclear pores in the surface of the nucleus, and they're just big enough to let RNA out because RNA is single-stranded, but small enough that it doesn't let DNA out because DNA is double-stranded. So that's kind of a cool thing. Here they show a sample of the endoplasmic reticulum. Now, in any other cell, if you look at it, this is going to fill up this whole area, the endoplasmic reticulum. But because this is just a cartoon version where they're just showing the individual things, they only showed you just a little bit near the nucleus. So you need to extend this out three-dimensionally coming out at you, and then you need to extend it all the way up to the cell surfaces. This is a picture of real endoplasmic reticulum, and it's taken with a transmission electron microscope. So they sliced open a cell, and you can see that it is completely full of this endoplasmic reticulum. And then scattered throughout the endoplasmic reticulum are mitochondria and other organelles. So a long time ago, back when they were discovering cells and discovering all these organelles inside of cells, women carried little purses that were kind of like today's fishnet stockings. So they had the you could have these as beaded bags or as crocheted with threads, and you had the cross pieces across there. But this is what ladies' purses looked like before, reticules. And so they named the transportation stuff, the organelle, inside of cells, they called it endoplasmic reticulum. So endo means inside, plasmic is outside the nucleus, it's in the cytoplasm, and reticulum would be a lady's purse. The endoplasmic reticulum, also known as the ER, because most people can't say that, is a series of passageways. So if I need to get something from the cell surface down to the nucleus, I can send it through the endoplasmic reticulum. Or if I have something in the nucleus that I want to get out, I can send it through the endoplasmic reticulum. So it's kind of a little bit like the blood system in our body, how we carry things throughout our body. So it's a transportation system. But if you look, there's smooth ER and there's rough ER. Now, also, when they used to first stain cells, they were little speckles all out in the cytoplasm. And they thought, well, it's just where the dye has left speckles. And lo and behold, it is actually a very, very tiny little organelle that kind of looks like, to me, it looks almost like a Mexican sombrero. So it's got a bulge on the top, and then it's got kind of a groove at the bottom. So these little hat-shaped structures make protein. So we're going to learn about messenger RNA and transfer RNA and ribosomal RNA pretty soon. But the ribosome is made of ribosomal RNA, and that groove is where the messenger RNA slides through, and as it slides through, proteins are assembled. So it carries the information, or reads the information, that is carried by the messenger RNA, and it tells it which amino acid to put where, and then it assembles it. Another interesting thing about the ribosome, it made us rewrite whole textbooks, because all enzymes that we knew of were proteins. And so that was in all the textbooks. All enzymes are proteins. And then we started looking at these ribosomes, and they clearly were doing enzymatic activity. They were linking amino acids together and making proteins out of them. And looking at it, taking it apart, we couldn't find the enzyme that was associated with it. And come to find out, RNA can act as an enzyme too. So that's why we had to rewrite the textbooks. Some of the ribosomes are just free. They're just out there, little dots, making proteins wherever they're needed. But some of them are actually associated with the surface of the endoplasmic reticulum. And so we call it rough ER because the ribosomes are attached. And if you watch, you can actually see proteins being made and dropped into the lumen or into the tube, the passageway of the endoplasmic reticulum. So that's kind of an efficient way to do it. Now, you can send it through the endoplasmic reticulum, and a lot of times it'll end up in the Golgi apparatus. So some guy named Golgi went through and he named a lot of stuff after him. There's Golgi structures in the brain, there's Golgi stains, here's the Golgi apparatus. But it's kind of like a coelomel. If you look at the ends, they kind of bulge. They're closed and they're bulged. So if you send something to the Golgi apparatus, it can just send it to the end and pinch the end off. And here you have a bag, a vesicle, pinched off, and it's going on its way. It can store something. It can go over to the cell membrane and do exocytosis, and it could exit the cell. So here it is spewing out the side. So this is probably either toxic waste that the cell didn't want, so it's taking its garbage out, or maybe this is a cell that is making some hormone or making some protein that's needed. So I'm going to guess that this one is dumping waste because this is a cell that absorbs because the microvilli. So Golgi apparatus is, of course, nowadays we don't do coelomeals anymore. We just use Ziploc bags. You want to put something and store it, you put it in a plastic bag and you zip it up. So in the case of these, it can just, because it's made of phospholipid bilayer, because it's made of fat, it can just pinch off. So anytime you see Golgi apparatus, you're going to see these spheres, these round structures. And some people call them vesicles, some people call them vacuoles. This one calls them vesicles, this particular textbook. One of the neatest organelles within the cell actually used to be a free-living bacteria. And a long time ago, this particular bacteria got inside of a cell, and instead of killing it, which is what we do to bacteria that invade our space, if they get inside of our cell, we kill them because we don't want to share food. We don't want them disrupting what we're doing inside our cell. But this particular bacteria, for some reason, was able to enter and live. And it persists in our cells today. And if you look in plants, it's also in plants. So this bacteria was so successful because what it can do is, in return for getting to be protected and getting to live inside of a cell and having all the food that it needs brought into it, and having the cell get rid of its waste for it, in exchange for all of these services that it's getting, like a hotel, it is making ATP for us. So it's gotten to where this bacteria no longer can live by itself. You can't take it out of the cell and have it live. And we can't live without it. If you give someone cyanide, the cyanide will kill the bacteria that are living inside of our cells. And unfortunately, because we can no longer make enough ATP, then we die also. So it's an endosymbiot. It's inside of our cells, and it's a symbiote because it does a really good job of making ATP for us, and we do a really good job of protecting it. So the name of this particular organelle is a mitochondria. And I get tickled whenever I watch Star Wars because I see Luke Skywalker and I see all these with their lightsabers. And they say the force is strong within them. And in one of them, they called them midichlorians. They said Luke has one of the highest levels of midichlorians we've ever seen. So the force is strong within the young Jedi. And I'm like, oh, they're talking about mitochondria. So here is a scanning electron micrograph. And if you want to look inside of something like this, you're going to have to cut it open a little bit. So they've taken a cell and sliced it open. And this is the mitochondria. So you can see they've cut away some of its surface so that you can see these folds inside. And that's their little ATP factories where they're making the ATP. Here's another one that they cut open right there. And if you look around, this is endoplasmic reticulum. You can see the ribosomes attached to it. So there's endoplasmic reticulum all over the place. So that's why I said you see it everywhere. And this looks to me like we're down looking at the nuclear pores of the nucleus right here. If you can see this part, you can see those little pores. Another kind of a mysterious thing that they found inside of cells, in animal cells, are centrioles. And they kind of look like bundles of shredded wheat, a little bit. We'll look at the structure a little more closely. But there is a set of them. And they wrote in the textbooks that the centrioles are responsible for cell division. And they help the cell to divide. And they're vital to mitosis. And so then the people who look at plants kind of tapped on the shoulder of the people who look at animals and say, you know, hey, our cells do the same thing that you're saying that the centrioles are doing, and plants don't have centrioles. So there's a wee bit of a mystery about what's going on with the centrioles, because plants have their cells divide, they do mitosis, but they do it without centrioles. On the side of the cell, we saw the Velcro-like structure, which is the desmosome, right here, with the protein extending across, extending, and then binding together right there to hold the cell. But you notice this isn't held all the way down. There's just a few of them to hold the cells together. And then at the bottom of the cell, you have what we call a basement membrane. And you have something that looks a little bit like the desmosome, but it's called a hemidesmosome. So it is, instead of holding a cell to a cell, it is holding the cell to the basement membrane. Right there. We're going to spend some time in just a second talking about phospholipid bilayers, or the fat that makes up the cell membrane, but that fat also makes up the Golgi apparatus. It also makes up the nuclear envelope. It makes up the ER, endoplasmic reticulum. It makes up the mitochondria. It's also phospholipid bilayers. So almost everything, those little bags that are coming off the Golgi apparatus, the vesicles that are being produced, all phospholipid bilayers. The ribosomes are RNA, so they are not phospholipid bilayer. And the centrioles, they were not exactly sure what they're doing in there, also are made of proteins instead of phospholipid bilayer.